Assalamu alaikum. In this video, we are presenting a group presentation. Here the course code is EEE 456 and the course title is Biomedical Electronics Sessional. The presentation is described by me and my friend Sumaya Begum Faiza and Shiab Ahmed. Our registration number is 192080002, 192081010 and 1920811 accordingly. The title of our presentation is UVC irradiation water treatment plant for inactivating SARS-CoV-2 replication. Now, I will start the presentation. The COVID-19 pandemic caused by SARS-CoV-2 has had a global impact on health and economy. Effective microbicides are needed to limit viral spread. UVC radiation is suggested as a way to inactivate virus like SARS-CoV-2. UVC interacts with virus by generating photoproducts that render them inactive. However, a reliable model for determining the UVC dose needed for inactivation is lacking due to variation in experimental conditions and measurements. Understanding the effect of UVC on SARS-CoV-2, considering factors like cultural medium, virus concentration, UVC irradiance, exposure time, and absorbance, will help replicate results in different labs. Accordingly, studying the impact of UVA and UVB on SARS-CoV-2 replication is relevant, given recent evidence of sunlight efficiency in viral inactivation. Now I will discuss the next slide. The importance of monochromatic UVC of 245 nanometer on SARS-CoV-2 and found that virus inactivation was easily achieved. Our experiments utilize a custom designed low pressure mercury lamp system providing an average of 1.082 microwatt per centimeter square over the illuminated area. The three exposure time were tested: 3.7, 16.9 and 84.4 millijoule per centimeter square, corresponding to different virus concentration. After UVC exposure, viral replication was assessed using cultural polymers chain reaction and analysis of SARS-CoV-2 induced cytopathic effect. Measurements were taken at multiple time points to track viral growth and perform complete various inactivation over time. The information is valuable for practical applications such as using UVC devices to disinfect surfaces and environments. The effect of the UVC exposure on SARS-CoV-2 replication were extremely evident and independent from the MOI employed. Dose response and time independent curves were observed. Now there are three figures are in the below. This figure will be described by my next project partner.
Assalamu alaikum. My name is Sumaya Begum Faiza. Now we can see here in figure 1 UV irritated SARS-CoV-2 replicated in Vero E6 cell in vitro. Virus titers were measured at 24 or 48 hours and also 6 days using solid copy number qualification. Viral replication was also assessed in cell lysis at the end of the 6 days culture. Duplicate cell culture condition were used. Their plot in panel D show virus copied normalized to untreated sample with mean values and the whisker representing the observed half range for described purposes. Also, we can see in figure 2 UV irritated SARS-CoV-2 virus replicated in Vero E6 cells in vitro. Virus titers were measured at 24, 48 and also 72 hours using real-time PCR. Viral replication was also assessed in cell lysis at 72 hours. Duplicate cell culture condition were used. Plot in panel D show the normalized virus copies compared to the untreated sample with mean values and whisker indicate observe half range for purposes. And also we can see the analysis of virus induced cytopic effect. No cytopic effect was observed in uninfected culture where E6 monolayers maintained in 50 ml centimeter square. UV treated complete medium for 72 hours. In viral infection of SARS-CoV-2, UVC untreated Vero E6 cell resulted in an evidence cytopic effect. Also, SARS-CoV-2 radiation with 7.2 ml UVC researched the cytopic effect induced by UVC antivirus. From the described figure, we can understand that controlled experiment model by researchers and then determine the UVC radiation required to inactive SARS-CoV-2 response to UVC radiation depend on the concentration of the virus. For low level, contaminate the closed environment and system from COVID-19 infected patient, and very small UVC dose of less than 4 ml like cm square was sufficient to fully inactive the virus. Even in the highest level, viral input concentration 100 mole viral replication was completely inactivated with a dose of 16.9 ml per centimeter square or higher. This finding demonstrate the high sensitive of SARS-CoV-2 UVC light. The results are crucial for design and developing effective UV based disinfection method to control SARS-CoV-2 infection. Now my group member Shihab Ahmed will continue rest of the presentation. Thank you all.
Assalamualaikum. I am Shihab Ahmed. Now I will describe in vitro SARS-CoV-2 infection assay. Vero E6 cells were cultured and prepared for a viral infection assay. The cells were cultured in DMEM medium with 2% FPS penicillin and streptomycin. SARS-CoV-2 virus had different multiplicities of infection, was treated with UVC radiation and willing doses. The virus was then inoculated into the Vero E6 cells and incubated for 3 hours. After incubation, the cells were washed and replenished with growth media. The cells were observed daily for cytopathic effect and viral replication in culture. Supernatant was assessed using a culture polymerase chain reaction method. RNA was extracted from the cell culture supernatant and cell lasted and then purified. Real-time PCR was performed using a specific probe assay for SARS-CoV-2 neurocapsid chain. The viral copy quantification was determined using a standard curve created from a positive plasmid control. The virus solution was illuminated using a low-pressure mercury lamp in a custom holder. The holder has a circular aperture positioned about 220 mm from the source, ensuring uniform illumination. A mechanical shutter initiated the illumination process. The virus sample suspended in DMEM was placed 30 mm below the aperture, and a single D well was irradiated from the top. The lamp intensity and spectral properties were measured using an ocean optic HR2000 plus spectrometer, calibrated against a reference source following NIST guidelines. The spectrometer featured a high sensitivity 2028 element CCD array from Sony, covering the range of 200 to 1100 nanometer with a 25 micrometer entrance slit and 1.4 nanometer optical resolution. The lamp intensity was measured at 5 positions on a 20 mm pro-sun after a 30-second warm-up. Here in this figure, we can see all 5 positions. Here the UVC line dominated the emission with a uniform intensity of 1.082 microwatt per centimeter square. The lamp remained stable within a 11E to 3 microwatt per square centimeter during the 130-second measurement. 3 exposure times 5, 23 and 114 seconds were chosen, corresponding to doses of 5.4, 25 and 123.4 millimeter and 123.4 millijoule per square centimeter. The effective doses reaching the virus were a interest, requiring calibration of the effective irradiance. They involved considering reflection losses at the air-water interface and the transmittance of the DMEM solution at 254 nanometer. The transmittance was determined from the spectrum in figure, accounting for weight losses. The spectrum was measured using a 1 mm thick water square and matched the solution thickness during UV irradiation. The process of water purification using UVC irradiation typically involves several stages. Pre-treatment. Water undergoes pre-treatment to remove impurities before UVC irradiation. This involves filtration, sedimentation, or coagulation depending on water source and quality. UVC chamber. Water passes through a UVC chamber with UVC lamp emitting lethal light for microorganisms. The chamber maximizes water exposure to UVC light. Exposure time. Water is exposed for a specific duration based on flow rate, UVC intensity and desired disinfection level. Higher intensities of longer exposure times increase virus inactivation. Monitoring and validation. UVC process is monitored by measuring intensity, flow rate, and other parameters for optimal conditions. Periodic validation tests ensure desired virus inactivation level. Post-treatment. After UVC irradiation, additional steps remove residual disinfection. This can include activated carbon filtration or chlorination as required. It is important to note that while UVC irradiation is effective in inactivating SARS-CoV-2, it does not remove other contaminants such as chemicals or heavy metals from the water. Therefore, it is often used as part of multivariate approach to water treatment, which may include other processes like filtration, chlorination, or reverse osmosis depending on the specific needs and regulations in place. To evaluate the impact of various UVC doses on the quantities of N1 and N2 copies, we conducted two-way A and OEA. This analysis focused on assessing the intracellular N1 and N2 doses in the supernatant. The dependent variables for the analysis were the UVC dose and the MOI. Additionally, to analyze the presence of N1 and N2 in the supernatant, we performed separate analysis for each individual MOI. In this analysis, the dependent variables were the UVC dose and the time elapsed. This analysis aimed to understand the effect of UVC dose and time on the quantities of N1 and N2 in the supernatant. Here is our reference. From this link, we will collect all our information to prepare this presentation.